detail. So now let's take a look at the structure of the sperm. So the sperm was discovered by Antony van Leeuwenhoek in the year 1677. So in the year 1677, Antony van Leeuwenhoek discovered the sperm. And then when we talk about the structure of the sperm and the size, so the size of the sperm is 2 to 5 mu micron in diameter, and it's about 60 mu micron in its length. Okay, so the entire sperm can be divided into 4 distinct regions: the head, neck, middle piece or the mid piece, and then the tail. Okay, so first let's see what is there inside the head. So the head consists of the acrosome. Okay, so the head consists of acrosome, and then it has also got the nucleus. So the head is going to have these two things only. One is the acrosome. So head consists of acrosome and then the nucleus. Okay, so you all know what is going to be the function of the nucleus. The nucleus is only going to be carrying the genetic material. Okay, so when we talk about the acrosome, so the acrosome, I already told you that acrosome is formed by enzymes which is produced by the Golgi apparatus. So these enzymes which are present inside the acrosome are called as sperm lysine. And there are two enzymes. Okay, so there are two types of enzymes basically. One is hyaluronidase and the other one is the proteinase. Okay, so these enzymes which are present inside the acrosome are very much needed for penetration of the ovum. Okay, so for penetrating the egg. So during fertilization. Okay, so during fertilization, the ovum or the egg has to be penetrated, right? So, and then only the nucleus of the sperm and the nucleus of the ovum can fuse together in order to form a zygote. So during fertilization, you know very well that the ovum is going to be having zona pellucida, corona radiata and all those things. So those layers are supposed to be penetrated and then only the nucleus can go inside. So how are those layers going to be penetrated? So the layers of the egg are going to be penetrated with the help of the enzyme sperm lysine, which is present in the head of the sperm. Okay, and then one more very important thing is that hyaluronidase. Why is hyaluronidase present? Because hyaluronidase, if you see, it has the ability to break up hyaluronic acid. So where is this hyaluronic acid? So if you see the corona radiata, so the cells which are there in the corona radiata are cemented with the help of hyaluronic acid. So this layer has to be broken. So how is this layer can be broken? It is with the help of hyaluronidase, which is needed for the penetration of the egg. Okay. So this is about the head region. So head is going to have the acrosome and the nucleus. Then the neck region. So in the neck, two centrioles are going to be there. Okay, so in the neck, two centrioles are going to be there. So one is the distal centriole. We have already discussed what is the function of distal centriole. It is only going to be forming the tail. Then the other one is the proximal centriole. So what is the function of proximal centriole? Proximal centriole is needed for the first cleavage in zygote. Okay, now just imagine that zygote is formed. Okay, so now this zygote has to develop into an embryo, right? So how is it going to develop into an embryo? For that, cleavage has to begin. So what is cleavage? You know, zygote is single celled. Okay, so cleavage is where the zygote will start splitting. So 2 cell, then it will reach the 4 cell stage, 8 cell stage, 16, 32. So like that, it has to start to divide in order to form the embryo. Okay. So the work of the proximal centriole is to initiate the first cleavage in the zygote. Okay. So cleavage is where the zygote will start splitting itself in order to form the embryo. Okay. So that is the work of proximal centriole. Now let's take a look at the middle piece or the mid piece. Okay, so middle piece, if you see, it has got a lot of mitochondria. Okay, so you know what is the function of mitochondria? Mitochondria is the powerhouse of the cell and it is going to be producing ATP. So why does sperm need ATP? ATP is very important for energy. Okay, so sperm requires energy to swim inside the female reproductive tract. So for that, the energy is going to be provided with the help of the mitochondria. And if you see, the mitochondria is actually arranged in the form of a spiral. You can see in the image here. Okay, so in this diagram, you can see that the mitochondria is arranged in the form of a spiral around the axial filament. Okay, so that is why it is known as mitochondrial spiral. So as the mitochondria are arranged in the form of a spiral, it is therefore known as mitochondrial spiral. Okay. And then moving on to the last region of the sperm is the tail, and tail is very important for motility of the sperm. Okay. So if there is any problem with the motility of the sperm, then the sperm will not reach the ovum and fertilization will not take place. So the tail is very important for the sperm to swim. Alright. So this is about spermatogenesis. In our next video, we will discuss about what will happen and what are the different types of abnormalities which are seen in case of the sperms. Okay. So I hope you understood all the concepts which we have discussed in our today's video. Thank you for watching.